Well, thanks for coming along tonight on a Friday night. This is a subject that I'm very passionate about, mainly because I think there's a lot of misinformation out there. But also it's a highly technical issue, and there's a lot of debate. Unfortunately, there's a lot of assertions that are made, possibly on either side. And one of the things that I want to try and deal with tonight is look at the evidence that we have now. So what is the scientific evidence that we have now that we can go out and reproduce in the laboratory? What are the measurements that we can make now? And the other thing is I'm very interested in a practical approach to things. My perspective is the knowledge that a man can use is the only true knowledge. The rest hangs like dust about the brains or evaporates like raindrops off rocks. So that was by an educational philosopher by the name of Froude, who lived back in the late 1800s and was the dean at one of the universities in Scotland. So there's a lot of irrelevant information like that is out there, so we want to try and focus it. Now, I notice I've published quite a bit in this area over the years, and it's something that I've been interested in, I guess, for a long time. So, you know, roughly, my background is that I went to the university here in Newcastle back in 1964. I enrolled, 16 years of age. I'd won a Commonwealth University scholarship and a BHP scholarship. I originally trained in physics. Applied mathematics. I did geology and chemistry as minors, and most of the physicists were ending up computer programmers. So I changed, went back and did the extra chemistry subjects, changed over, and I topped chemistry at Newcastle Uni. So my name's there on the honour roll in the chemistry building there. Subsequent to that, I won the highest chemistry scholarship in Australia that was offered at that time, the Tychoxide Research Fellowship. And I went to the University of Tasmania because at that time Professor Harry Bloom was a world famous chemist and probably, yes, one of the most renowned chemists in Australia down there. I did a master's degree in chemistry there. I won a minor prize for my presentation. But it was while there that I became a Christian. And when I became a Christian, I was working as a research fellow at the uni. People would ask me, well, what about evolution, and what about the age of the Earth and this sort of thing? So I began reading in this area. I began reading up. I had a colleague who was doing his doctorate in geochemistry at the uni as well. He had a European shovel handle dated. He was working on a gold field in New Zealand and studying the geochemistry there of the gold mercury deposit, and he'd had a prospector shovel handle dated by radiocarbon, and it came back at 6,600 years. And he thought, this is crazy, you know, the oak tree that this was made out of can't be that old. So those sort of things, we began reading up different papers. So 71 was over, that was in 1971 I was baptised. So that's over 40 years ago. So I was looking at that. And I became very interested in the research. So I went back to uni. This time I read for a doctorate in the area of epistemology as related to the biomedical sciences. So how can we know? So one of the problems we have in the biomedical sciences is the problem of synergy. It's very difficult to predict drug outcomes and these sort of things because of the complicated biochemistry. So are there any better ways of knowing than the reductionistic method? So that was one of the things that I looked at, and so for my thesis I actually had to review the theory of evolution's Darwin origin and how we know the different methodologies that are used. So that's my area. So I taught physics and mathematics to engineering students for many years. And while in Tasmania, I set up their for technical and further education, their applied microbiology courses and their medical laboratory technology courses. Then I joined the company that I'm with now, Sanitarium, as their chief chemist. I notice on some of the websites, some of my detractors tend to put down food chemistry. Well, food chemistry is extremely complex. I mean, if you're analysing rocks, you're analysing forensic, that's pretty straightforward. food is an extremely complex matrix. It is just so complex. And now, of course, we're discovering that there are many minute, very minor compounds in our foods that upregulate genes, downregulate genes, involved in, protecting against cancer, accelerating cancer, extremely complex. And of course now we're getting into even more involved areas with the understanding of how the gut microbiome works and how our gut bacteria send signals to the brain and all sorts of things, and how these signals are modulated by compounds that are produced by bacteria. And this is really fascinating. And then, of course, extending, you've got a vaginal bacteria, you've got a seminal biome, a vaginal biome, and these, now we know, can affect the intelligence of the child that is born. So it's really, really fascinating. This is an area that is speeding in terms of research very rapidly. We're discovering, how can this be? What is happening? And it's all to do with the compounds that these bacteria produce and the signaling effects that they have. And then, of course, there's the virome, the viruses that live inside us. And then there's the fungi that live inside us. So there's lots of exciting areas. So to think that food chemistry is, formulating hamburgers is a bit, limited in its view. So tonight, let's have a look. We've divided what I'm talking about tonight into a couple of sections. The first section, I want to look at the chemical origin of life. What would be involved? How did life begin? Where did we come from? And then the second part we'll look, after a bit of a break, is the part that relates to Darwinian evolution. How did the different types of animals form and different types of things form over the ages, and is that possible? And have a look at the actual mechanisms that are involved. So I better go to my slides now. Let's see if this works. Whoa. So how did we get here? So this is, chemical evolution. If you read most textbooks, they'll say that the Earth was formed about 4,500 billion years ago and life arose thereafter. Or other textbooks will say life arose about 3,500 million years ago to 3.8 billion years ago, somewhere in that period. But they're just assertions. They just state that. They don't know. No one has a mechanism to date for how life formed. So why is this so? So textbooks assert that this happened. Scientific American articles, there was an article came out a couple of years ago with a professor of genetics at Harvard. And again, that's the assertion. Life began. And if Richard Dawkins, when he was interviewed, how did life begin? Well, maybe it came from outer space, which really doesn't solve the problem. So an attempt to explain the life on Earth was developed by Dean Kenyon, who was professor of biology at San Francisco State University. And you can look up now if you go on YouTube, and he came to realise that the code and the code reading mechanism couldn't arise by chance, because the code's useless without a code reader. And the code reading mechanism is very complex. And then you also need the little factory to interpret the code reader and actually synthesise the proteins and the compounds. So for all these things to arrive at the same time was a bit of a problem. And so there, if you just Googled the Charles Darwin of our time or Dean Kenyon, there are a whole lot of YouTubes that will come up that where he will talk about why he changed his position that chemical evolution is absolutely impossible. So, and I'm happy for this to be made available. So we have Francis Collins here, who led the genome program. Again, while he is an evolutionist, he believes that the origin of life must be supernatural. God must have created life in the first place. So another guy, another philosopher, a philosopher of science, educated at Cambridge. That's the book that he wrote. It's quite a thick book. It's quite detailed, but essentially he argues that the DNA code is so complex that it could not arise by chance mutations. And we'll have a look at this a little bit more. So And there are many other, biochemists that I could list. Was talking to a colleague last night who at Sydney University and who heads another research unit. He was saying on some of the forums now, this is a number of the guys are saying, look, our data just doesn't fit evolution. So living cells are amazingly complex. So for a living cell to form, and we have to form a large number of biopolymers. So these are quite long molecules that have to be synthesised from smaller molecules. Now that generally happens by the removal of water. But we'll come to that in a bit. We have to have coding information. We have to have a code reader arrive. We have to have protein production mechanisms. And we have to have, and this is an important thing that's often overlooked, hundreds of chemical reactions in a state of disequilibrium. Just the right amount of disequilibrium, and I'll explain what that is in a moment. So we can see that these long-chain molecules are your proteins and your sugars and your fats and so forth that have to form. So certainly there have been experiments, controlled experiments in the lab where they've formed some of the simple, very smaller molecules, but they've been unable to form by some sort of natural process these long polymers. Now, one of the reasons that is because these carbon chains, when we form the polymer, it involves the removal of water. And most of the theories that are proposed are that life arose in the seas, and that is biochemically impossible because of Le Chatelier's principle. It would drive all the reactions the other way. Polymers break down in water. And so the chemistry points the other way. So that's why there's been attempts to propose theories that maybe this occurred on the surface of a clay particle that somehow catalysed it. But again, these are all theories. There's no proven evidence. There's no research papers with substantiated mechanisms showing how the first DNA code could form. And of course, you not only have to have the DNA code, but you've got to have these long proteins, the long sugars, the long fats forming. So we've just got a diagram of a cell here, and you can see there's a DNA code, there's a membrane, there's some plasmids, that's a little bit of DNA code. We've got a little ribosome, which is a factory. Right? And we can draw these things, but if we blow it up a little bit further and we just look at that envelope, we can see that there are a whole lot of these very specialised molecules that have to be synthesised and formed just to form that membrane. Now, the code for all those things is encoded in the DNA to make them, plus the code reader. So you've got a real chicken and egg problem. They protect the code, but they're made by the code. So we've got massive, massive problems this way. We've got massive problems in terms that the codes for every one of those molecules has to be encoded for in the DNA. If we go to a little bit more complex view of the cell, we can see there's more little factories in the cell, factories that process fats and processes that make energy. All the codes for all those factories are in the DNA code. It's an extremely complex code. Scientists can't write a new DNA code to make a new type of cell that works. So with our best intelligence, we can't write new code. And yet evolution requires that there's somehow this amazing code formed by chance. There we go, just another more complex cell again. So, and some of the components of the cell. So all these components have to are encoded for in the code. And we know that the smallest cells contain millions of molecules. So not only do you synthesize one molecule, somehow randomly by chance when it first forms, you've got to synthesize millions of molecules. Some of them identical, many of them identical to another, about say 600 different types of molecules, in millions of molecules quantity form by some random process, but all in the same location so they could somehow come together. We know that all this scenario, it's just impossible. We don't observe anything like that happening in the laboratory. And so we can see even in these simplest cells, that we know of Mycoplasma genitalium. Now, there is one, if you get pedantic, that is smaller, but it needs a host organism. But we've still got 580,000 pieces of code arranged in 471 genes. So it's extremely complex, and so it's imagining, 5 large textbooks, the writing on that just somehow forming by natural process, raindrops falling on ink or something like that. We know it just doesn't happen. You don't write codes like that. They don't form in nature by themselves. And these codes are complex. They have to be precise. If the code isn't precise, the organism doesn't work. It either dies or becomes diseased. And so some of these probabilities have been calculated by a number of mathematicians. Back in the 60s, there was a large mathematical conference, I'll refer to it in the book here, where they looked at evolution. They said, look, from a mathematical point of view, it's absolutely impossible. But the biologists continued on. They said, look, you mathematicians don't know anything about biology. But of course, since then, many other people have looked at it and echoed the same thing. From a probability of the biochemistry occurring, it's absolutely impossible. So let's have a look. We've got here a representation of the DNA molecule and the little code, the different parts of the bases. So you've got a phosphate sugar backbone and then the different nucleic acids across there. So if we look at the single E. coli, we've got about 2.5 million protein molecules made up of 4,000 different types of proteins. So for that cell to form, you've got to have the codes that write all and form all those different types of proteins. And you've got to encode for them. And we can see that there also in the cell is a huge number of sugar molecules there. There's many different lipids, over 100 different types of fats in the little E. coli cell. And there are 800 other different types of compounds. So you're not looking at just simple biochemistry, just involving a few compounds here. We're looking at a large number of biochemicals that are all present in exactly the right concentrations. If the concentrations get slightly out of kilter, the organism dies. And so these are the genes that are responsible for some of the functions. So when E. coli, its genome was first determined, these were the number of genes that they were able to identify, those involved in amino acid metabolism and different cell envelope and so forth. You can see the number of genes listed there. And in energy metabolism and so forth, replication, the number of genes that are involved, there's about 4,500 genes. And so at that time, our chemists could only figure out the function of 1,500 genes out of the 4,000 odd genes that were present. And so the complexity of just this simple little single cell organism is absolutely enormous. And yet evolution requires that all this complexity arose as a result of just random chance chemical reactions. And we know that that is absolutely impossible from a biochemistry point of view. We don't observe that happening in the lab. So the other thing is that even if you form the DNA code, you still have to have a code reader at the same time, also forming. And this involves, again, complex molecules which unwrap and copy the DNA code. And this is an amazing machine. There's a video that has been developed by researchers at the Walter Eliza Hall Institute, Division Cancer Research Group at the University of Melbourne. And so there's a schematic of it. We can see the DNA. The DNA is unravelled. Then a copy is made, the RNA, which goes into your little factory here, your ribosome, and which reads your code and spits out the amino acids. So it's a little miniature factory. It's absolutely amazing. And we'll just watch a little video of this now just for a couple of minutes. So this
using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules. What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Is there another one, is there? Okay, yeah, we'll watch this.
When the
Better than me
RNA copy
talking.
is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Okay, so just think about this. The code to make all the components of that system, according to the chemical evolution model, has to arise by chance. Now, comprehend that. The code to encode for all those mechanisms, because all those mechanisms and those components are encoded for in the DNA. And that's why Eugene Koenen, one of the top biologists at the National Institute of Health, has said we've got major problems. We've got major problems with the theory of evolution, and particularly chemical evolution. How can that mechanism arise? How can that mechanism arise by chance to read the codes, to form the components of the cell? It's extremely complex, it's elegant. Let's go on. So Meyer, in his book, looks at these polymers, and he actually published some papers that to show that from a mathematical point of view, it's absolutely impossible for these components to form by chance. The paper can be put up there. It was originally published in a mainstream journal. They asked for it to be withdrawn. So but you can read it there and judge for yourself. So the two basic premises that for life to come here, and some people argue with me and say, well, evolution doesn't deal with the origin of life, or that's a biogenesis. Okay, well, the big picture is how did we come to be here? So we had to start somewhere. And we'll talk about the other part in the next stage. But we've got a biogenesis. Somehow, a living organism, like what we've just seen, had to form. And those components, that mechanism has never been observed in the laboratory. As I said, we can't even write it. But there's furthermore, say we took a little E. coli and we dropped a tiny little drop of toluene just on its outer membrane, that the cell would die, right? Because we disrupt energy transfer just for a moment. And what happens then is we have all the components there, right? We've got all the biopolymers, we've got all the connected metabolic pathways, they're all there. But the cell is dead and we can't make it alive again. Why? Because we can't put hundreds of chemical reactions simultaneously in a state of just the right amount of disequilibrium. So again, the probability of that happening, we know that would be, when things die, they can't be made alive again. If they do, we call it a miracle. So either way, you've got to have a supernatural origin of life. So to this day, step one has been achieved in the laboratory by scientists, but not in nature. To this day, it has not been possible to perform step 2 in the laboratory, despite the best efforts, let alone steps 3 and 4. So here again, we have experimental evidence on the basis of what we know today, that's impossible for a living cell to form. We know from our understanding of mechanisms how living cells operate, that's impossible to form. We know mathematically from a probability statistics it's impossible to form. George Shea, reformed professor of biochemistry at the School of Medicine, Loma Linda University, he said that the proponents of chemical evolution are choosing to stick with a simplistic 80-year-old model against a background of a tidal wave of evidence that abundantly shows the mind-boggling chemical complexity undergirding life. He goes on to say, our inability to bring back to life intact but dead E. coli cells in the laboratory shows the impossibility of spontaneous generation of life. And that is, even if all the components of a cell were there, it could not come to life by itself. Life requires a miracle. So in the next presentation tonight, we'll look at why all the different types of life on Earth could not evolve by mutations and natural selection. Now, some of you, for a lot of people, this is, a bit hard to swallow. I have put a lot of data up on Facebook. It's Facebook/Evolution Impossible with links to research papers by world-leading scientists and philosophers who are raising these questions about this. So, for example, you've got Thomas Nagel, Professor of Philosophy at the University of New York, with his recent book, Mind and the Cosmos. I think I'll talk about that in the next talk as well. Why neo-Darwinian evolution must almost certainly be false. So, and we've got a lot of people that are in this area, like Dean Kenyon, who wrote the original chemical evolution textbooks. He realises now that they're impossible. So there are many scientists in this area now. So anyway, I hope that stimulates some thought. We'll have a little bit of a break now, and then I'll do the second half of the presentation, and very happy to take then questions at the end. And I'll attempt to answer them as best I can, or refer you to literature that you can look up. Bear in mind that in the book, I've attempted to use the best peer review literature that I can, so from good sources, the top journals, top research journals. Okay, well, have a bit of a break and enjoy some snacks.